to introduce our first speaker of today, Guy Cochran. Guy is team leader for data coordination and archiving and head of the European Nucleotide Archive at the EMBL-EBI. Since this year, he also is the founding executive director of the Global Biodata Coalition. Guy has received his PhD in cancer cell and molecular biology from the University of East Anglia in 1999 and carried out postdoctoral work in the molecular biology of photoreception at Cambridge University prior joining EMBL-EBI. Guy has over 2 decades of experience working with and leading biodata resources, and he is an authority on large-scale international sequence data sharing. He led the adaptation and extension of the European Nucleotide Archive for the purposes of rapid global pathogen data sharing, which is better known as the European Covid-19 data platform. And I would say, Guy, the stage is yours. Thank you very much for joining us here today.
Great. Thanks very much for the introduction, Antje. Do you see a full screen slide and do you hear me?
Yes, we hear you and the slides are perfect.
Excellent. And thanks very much to Euro-BioImaging for hosting. I'm delighted to be here. So I think there's very little doubt amongst many, certainly not on my side, about the value that there has been from having fair and open data during COVID-19. So being able to discover data sets of relevance, being able to access and to reuse those data sets openly has been important in accelerating the world's response to the disease, and also accelerating and deepening our response, that we can respond better and move further and faster. So I've got a few examples here just to start off with. And the first one is that prior to the emergence of SARS-CoV-2 as a new species, there were already some 32,000 different sequences from other coronaviruses that were publicly accessible and discoverable. And actually, without having that context, our understanding of the biology of the life cycle of the weak points, the target points for SARS-CoV-2 really would have been retarded. So we had a very important head start because there was pre-existing context and pre-existing understanding. If we move on to vaccines, so the vaccine programs were triggered very rapidly, were very impressive. And on the right here, we're looking at a sequence succession, so an openly available, discoverable sequence that really informed the early stages of vaccine design. Without that, of course, things would have been much slower. When we get to treatments and therapeutics, having data available from, for example, structural biology, having data available from a chemical biology was equally important. So here we're looking at some RNA polymerase structures. So RNA polymerase critical to the life cycle of RNA viruses. Here we're looking at some nucleoside analogs, and these were openly available data from other species and from different compound libraries and so on. But what they enabled the world to do was to understand existing therapeutics that were available, which may be prioritized and may be appropriate for repurposing. And this is the route actually that one of these blockers took, the molnupiravir blocker that's used against COVID. And then of course, we're perhaps all very familiar with the variant analysis work where surveillance programs and sequencing of viral samples, viral isolates from patients with COVID really has led to an understanding of the connection between different parts of outbreaks, different parts of the pandemic, the changes over time, the emergence of variants. And on the right here, we're looking at a very rapid third-party analysis of the fairly and openly shared data that first emerged on the Omicron variant late last year. So it was with that kind of concept in mind. So we didn't know exactly what was going to happen all those years ago when data were being made available and shared, but we knew that these would be important. So with that kind of ethos in mind, we realized at the beginning of 2020 that we had a job to do in bringing together the existing data sources that would have data of relevance to the response to COVID, but also to enable the capture and the preservation and the presentation of new data that will be relevant. And so that's really what became the European COVID-19 data platform. And it still persists, and it is there to provide open and rapid access to data, tools and workflows that are useful in the scientific response to COVID-19. So the system builds on open standards. And I like to think that it also contributes to those open standards. It's European by name, but it's global in terms of the data that it covers and the access. So it's access from people all around the world. We were able to launch quite quickly. And really what we are is a platform, a foundation for people to make an informed response to COVID-19 in all the broad senses that can have. So there are a number of components of the platform, and I'm going to step through each of these in turn and outline them. We have technical components that deal with viral sequence and human data. We have integration systems that link things together. Then we have the COVID-19 data portal, which is the most visible part to most people. This is the entry point for most people into the different data and tools. We have a lot of engagement work and coordination work that continues that's still ongoing. And then we have a new thread, which is substantial, which is about preparedness. So this is taking us beyond SARS-CoV-2 and beyond COVID-19. And so before I start going through the components, this is my acknowledgements plus foundations background slide as well. So the platform is a huge partnership between many different groups, and I can't mention people by name, that there simply isn't enough time. But I'm going to talk about the key partners in their institutional or project groupings. And so on the top left here, we have my institution, the EMBL European Bioinformatics Institute, which for many decades has provided data resources around molecular biology and around the literature, and certainly has been capturing global molecular biology data from many different sources, including around pathogens for some time. Bottom left, we have Elixir, which is the bioinformatics infrastructure. So here we have a ready-made network of bioinformatics experts and capacities around European countries that's played a very major part. Top right, we have the European Open Science Cloud. So this is really our link into the different research infrastructures, including, of course, Euro-BioImaging. So this has been powerful in helping us to coordinate and link across. And then here we have many different institutions that have been we've been working with on the EBI side for well, probably over a decade now. Public health institutions, academic institutions, technical institutions in building up pathogen biology related data resources and services. And then of course our partnership with the EU is really important, and we now have support from some 6 different, or have had support from some 6 different European Horizon grants. So first component is there for people who are working with viral sequence data. And so this is one of the major components. So what we call it a SARS-CoV-2 data hub. And a data hub is really a toolbox for those working with viral sequence data in some way. So it allows people to upload and to validate and to QC their data, to share, to publish their data. It's linked to cloud compute capacity with different workflows around assembly and variation calling and so on. And then there's a host of different exploration and visualization tools so that people can interpret the data. And there are some screenshots of different tools. So here we have one of the rapid upload tools. This is a data management control tool, some QC there, and then some different, I think these are variation exploration tools. And so what we did, and I guess we continue to do, is to go from country to country, really to understand the layout of viral sequencing, what the national programs are, and to help to put in place the right structures, which may be brokering structures or direct submissions to allow the data to flow into the central system using the data hubs at national or at institutional level. And so this was initially work going from country to country and understanding the layout. And it's been productive in terms of the mobilization of data. So we now count something like 5 million isolates worth of raw data or assemblies each from the virus. So it's a significant body of data. And we've been able to use it. So people have done their own analysis, of course, and made their own interpretation. But we've been able to use this for the systematic analysis of large data sets. So we now have an analysis running that's covered more than 2 million different viral isolates, where we're using, because of the fairness and the standards compliance and the interoperability of the data, we're able to apply consistent analysis workflows over the data at scale and produce some outputs. And so down here in the middle, public health reporting. So what this analysis leads to is a set of publications or bulletins that we send out under the VO project, one of our projects, as a variation report. And this goes out to many different public health agencies and other stakeholders. And essentially it's a list of happenings in the variation space, interpretations and so on, that we hope is useful for public health players. So onto the human data component. So this is dealing specifically with the sensitive human data. So data that can be revealing of a patient's privacy, where controls are required on access. And so here we build on top of the European Genome Phenome Archive, which is the established data infrastructure for sensitive data from humans, particularly around molecular data, particularly sequence data. And in there, we've, from many parts of the world, 10 different countries, we've captured something like 500,000 individuals worth of data, so research participants worth of data in 32 different studies. And so there's a variety of different data types, sequencing, expression, and so on. And we're working as part of the COVID-19 host genetics initiative to have a global coverage. And there's a comparable database over in the EMBL system over in the US. But I think a further important element of the COVID platform is that it's been able to accelerate work on the federated system. So as you all no doubt know, although there are ways in which sensitive human data can flow across national borders, say within Europe, actually it's often very difficult to use those methods, and there's a strong preference for data not to be traveling across borders, so for data to remain within the jurisdiction and stay within the control of the countries themselves. And so what the federated EGA does is it builds out a structured federation of different nodes where the data can stay local, they can stay stored at institutional or national level, but there is a set of systematic connections and systematic interfaces that one can go in anywhere in the system and find the content that one is looking for, so make the discovery of data, and go through a consistent set of interfaces for putting in an access request, for describing the study and describing how the user will comply with the requirements of the access conditions. And then putting in that request, and then when the data, if access is granted, being able then to go to those data sets through systematic interfaces. So the data stay local, under local control, but there is metadata service, and there is discoverability across the whole network. And if you look at the map here, so we have countries all over the world who are now moving forwards towards providing nodes within the EGA Federation. And there are 5 European countries that are now very close to finalizing their connectivity. Many of these are existing databases, but finalizing their connectivity with the federation. And this will be, I hope, a long-lasting and very powerful implementation, not just for COVID, but well beyond. So onto the integration component. And so here I'm focusing on sample level integration, and I'm looking at the human pathogen divide. And so one of the important but really tricky connections to make between different data points when looking at infectious disease is the connection between the pathogen, things you know about the pathogen, and things you know about the host, the person or the animal was infected by that pathogen. And in some cases, I'll have an example later where one has a vector in between. So there's a vector that carries the pathogen as well. And so the interplay between the genetics and the other characteristics of the different organisms involved is really important, but in data terms, it's been quite elusive. And so our sample-centric integration really takes the concept that if we take the example of a human participant in a research study or a COVID patient is effectively, in our view, a sample, but we have a proxy record that ties everything together. We may not know very much about that patient, and we may not be able to reveal anything, but it ties things together, so it's an identifier. And then against that, we can have different samples. So if a blood sample has been taken for genotyping, we can capture that. If a swab has been taken for viral sequencing, we can capture that as well, and we can keep them all tied together. And so on the left here, we're looking at the different levels to which we can, through the platform, capture different types of data. We can have very deep private data sets themselves in EGA or in federated EGA, but it's not always possible to do that. Perhaps we can capture the information at the level of the samples that are taken. Perhaps it's only at the level of the whole cohort of patients and their samples. Perhaps it's just an overall description of the study. But so we have many options for capturing the data, but fundamentally we can use this concept of sample to link things together. And so here's an example of how we've done that. So and this is an example from the Netherlands through the Recoded project. So if you look at the top left here, what you're seeing is just a bit of a screenshot of our cohort browser, part of the web portal. And this is a study, so this is a cohort, a group of patients. And we have a tick here and a tick here. So this is a tick that shows we have viral data, a tick that shows we have laboratory data from the human as well. And so here is the sample. This is the proxy sample for the human. It doesn't say much about that human. It doesn't reveal very much, but it's sufficient to show, to give access to serology data. So this is a serology data set that's linked, and it's sufficient as well to give access to the viral sequence data and the viral genomic data. And so this all hangs around the central notion of a sample. And in this particular example, we don't yet have case report data, but one I will hope to be able to show in future. There's also case report data, so deeper clinical features of that from the patient that was infected, but those would be behind controlled access, of course. So onto the COVID-19 data portal, which is really the entry point for most people. And this is a data portal that brings together all the different data sources of relevance. And importantly, it's a portal. We're not building new databases here. We're really using existing infrastructure. And I think this is one of the reasons we were able to move so quickly. So overall, we have something like 12 million different records, and these span the omics platforms, other molecular biology platforms, the literature, and so on. And so the viral sequence I was mentioning, the big numbers are here. The host sequences I was mentioning are there. The literature grows very rapidly, and then many perhaps more reference data sets in proteins, structural biology, and pathway network systems biology data, and so on, and then information about different samples, cell lines that can be used, and importantly, there's an imaging section. So we've been able to index the imaging. So this is Bioimage Archive, the repository. We've been able to index in the same system imaging data, and we hope this will be useful in future as well. And so here we're looking at a few screenshots of different parts of the website, and I've just put in these are thumbnails of electron micrographs. So that's very sort of fine-grained imaging, but we have some low resolution stuff of tissues and cell lines behaving in different ways, for example. So here you have access to the different data sets. You have access to tools that operate on those data sets, and you have a set of associated APIs. So everything you can do on the web, you can do programmatically as well, and then download and management tools and so on. So do go and have a look at the portal. It's growing all the time, and it has some very useful data sets and functions, I think. It's heavily used, as you all know. One of the features of data, certainly of open data, is that actually you don't get to know directly what it is that people are doing with the data, because you don't quite know who they are, which is expected. But here's just a few anecdotal examples of how the data are being used. Overall, we know that there are many users. They're coming from many different countries in the world. And so here we have chemical biology meets candidate targets on the human genome. So candidate therapeutic targets on the human genome. There's a prioritization tool provided by Open Targets. On the top right, we have the Galaxy system, which offers a whole host of different workflows for COVID-related data that's connected to the portal and data in the portal. This is from Barcelona, CRG. This is a viral variation navigation tool that allows you to explore in very fine detail the variation data from the virus. I've mentioned the national data integration and the public health reports, and this is the NextStrain system. So this is a Swiss system for looking at viral lineages, for example. So there are many other examples of how people are picking up different data sets from the portal, and they're adding value in some way and presenting these again to further users. So onto the engagement coordination part. Here I can just show a few examples. And this is a group we call the national coordinators. We tried to put together key stakeholders involved in the COVID response from health, public health, from research, but also from funding bodies, from policy making groups and infrastructure people and so on. And we've had a host of different meetings and discussed many things, including, of course, data standards, fair data, data openness, and so on. And much of this has branched out into specific discussions in different areas. On the right here, we're looking at the national portals, which has been a really nice development. So we have the central COVID-19 data portal, and then now 10 different networked national data portals. The function of these is that they provide a national view for a scientist working in a country to find out what's happening nationally. So the funding opportunities, the collaborative groups, the compute provision, the services that are available locally, but then to move smoothly into the central system for access to those data or those metadata that have been centralized. So that's been a very nice development that's emerged. A lot of our work has been about encouraging openness in data and good practice in that openness. Right from the beginning, there were discussions about how open the data should be with all sorts of different groups. Currently, the discussions are focusing in particular on various WHO consultations and the meetings of the G7 and the G20, where this is also data sharing for pathogens is also a discussion. But we've done various things. Some examples here is an open letter from a while back, and then much more recently this year, this is an opinion piece that really tries to argue the value of research reuse of data that are generated for public health purposes, and the reciprocity of value, where public health needs access to research outputs to be applied better, or to use genomics better. So I step outside the narrative just briefly to raise the Global Biodata Coalition. This is a group, an initiative that I've been, as you heard, I've become involved in recently. But I think it's very important here, and it's good to raise this, I think, with the imaging community. So the Global Biodata Coalition is a coalition of funders, of research funders, who get together and realize the absolute importance, how essential the data resources are, so databases for life science, how essential these are for the research process, how we rely on them every day very deeply to deliver our research, yet recognizing that actually the coordination, international coordination of many of these resources and the funding for those resources is really often not very sustainable and very planned out. And so some of the databases that the world relies on enormously have very short-term funding plans and actually are at risk. And so the funders get together to try to address this. And really the work is about aligning the funding mechanisms with and allowing the research funders to offer in their programs appropriate forms of funding, and then aligning the data resources and the managers of those resources to that form of the programs that are now available. We're currently working, and you may have seen a call for global core biodata resources. We're working on an inventory of all of the data resources. So actually, a lot of the work is finding out what the data resources are, how they function, how they interconnect, and what's really most important, and what is most at risk. And so a lot of our work is about profiling and landscaping all of that. But in that landscaping, we absolutely need to be capturing image informatics, image data resources into that equation. Then the rest of the work is, some of the rest of the work is about understanding funder views on sustainability and what they might put in place, and understanding open data and policy and so on. So you'll hear more about that, but I do encourage you to get involved and find out what's going on there. So just finally onto the final component, which is newer, which is preparedness. So how do we take what we've got, what we've built from COVID-19, from our platform, and reapply it for future diseases, emerging and re-emerging diseases? And I just got two examples, and this is a project really, so we collaborate on this project. It's led by Frederic Bartimeus in Spain. It's part of VIO, and this is Mosquito Alert. So it's a mosquito, an important vector of disease with its distribution patterns moving into places where disease-carrying mosquitoes previously weren't. And it's a citizen science project really to monitor the mosquitoes at this point. There's no physical sampling, and there's no molecular or deeper imaging. This is just identification at the moment, but there's a potential for this to lead to other things. But certainly it's happening at a large scale. And I'm delighted to say, so this is an Asian tiger mosquito, and I'm delighted to say that we're feeding those images into the Bioimage Archive as well. So we're capturing those into the underlying infrastructure for future connection and reuse. And then finally, really our first opportunity to directly repurpose some of the components that we have. There is, as you will know, an outbreak of monkeypox virus around the world. We hope that it doesn't go very far, but we will make a response that's appropriate, or that's proportionate to the in proportion to the severity. So this may go a bit further, it may not. But here we've been able very rapidly to reuse some of the components, and so certainly providing submission tools and submission guidelines. On the right here, you see a web view of this is a tab on one of our web portals, but it provides access to the sequencing parts of the data set with some searchability all in one place. And we're providing data hubs, and we're working on a computational analysis workflow to process and to extract, for example, variation data from monkey virus genomes. So we will add more components if it's required, but it's nice to see that at least what we have is with not so much work, is directly reusable. Anyway, I leave it there. I thank you for your attention and look forward to any comments and questions. Thank you.
Thank you very much, Guy. That was a really, really great presentation, and gave us a very good overview of what's happening with the COVID-19 data portal. Myself, I have several questions, but let's first open the round to the participants here. So please raise your hand or just speak up in case I don't see you, to ask your question to Guy. Okay, as so often, people need some time to think about this. So my first question would be, Guy, I had the honor about a year ago to present some of your slides at an Omero user forum, as the Omero team here might remember. And at that time, I was reporting about 3 million data entries. Today, we learn it's far above 12 million data entries. At the same time, we all know that people are tired of the pandemic, less people go for testing, less PCR testing. So what do you predict? Where's the COVID-19 data portal going? Is it still going exponentially up in getting new data sets, or do you also see a curve slowing down?
So yeah, that's a really good question, Antje. Indeed, we're seeing the curve slowing down. The dominant data type that feeds those numbers is the viral sequencing from sequencing of positive cases, and there, in a number of parts of the world, there's been significant reduction of the national programs, and so we see a corresponding. The numbers are still increasing, but we see a significant drop in rate. And actually, if you're on the portal, there is a statistics under about. There's a statistics page, and you can see things curving off. So I think we continue to get data, but I think it will reduce. And I think unless there's a major or a new variant that has major importance, that triggers some need to do something very specific and to do deeper sequencing, I suspect that we're not going to go back to the rates we saw perhaps a year ago. That doesn't mean that we have to finish, though. I think that you don't necessarily need all of the data to find patterns. We've already seen that. But I think being comprehensive and seeing it through to the end, well, I don't think there won't be an end, but seeing it through for a long time to come will be very important, because at the end, at some point in future, there's a big retrospective, I think, that says, well, like now we know what we know now. Can we learn from that with all of the data that we've accumulated? Can we learn how better to respond? What it is we can do early on in a pandemic that's similar? What can we do better next time? And so this big retrospective, I think, needs comprehensive data sets. So we carry on. And actually, of course, at this scale, the system is automated. We've been able to, for the data flow, we've been able to set up very good, I didn't have time to show the slides, but very good arrangements with different countries who broker data into the system. And those networks carry on operating, and they will also be extending, I hope, to other diseases and other pathogens. And so it's not zero effort, of course, to continue operating. But I think it, over a scale of a few years, it may become something that's a little bit more background. One still collects all of the data, one still has the views, but perhaps fewer new developments and fewer new connections into the system. But I don't know. I mean, it's still raging. The disease is still raging around the world. So there could still be surprises.
Yeah, I think all of us these days attending in-person meetings get every morning some bings on their phone that another person they just met has tested positive. So yeah, it's not over by no means. being a little bit self-aware or even self-critical here, looking at the image data sets in the COVID-19 data portal, I think the imaging community, we have to ask ourselves, why do we have only 29 data sets there? Of course, you say that the PCR testing is the major driver behind the millions of data sets that are there, and we never can compete with that. But do you have any advice for us how we can mobilize even further? Because we're trying really on that, and Aastha might talk about this in a minute when presenting the Bicovit project. But from your perspective, Guy, as the original father of the COVID-19 data portal, what is your expectation to this community?
Well, so I think the numbers, I mean, the numbers are useful to track changes and patterns, and certainly tracking, the numbers of sequences has been a really important tool to help to know where we should focus efforts. But they're not necessarily comparable, and what I mean, does one expect imaging platforms to produce huge numbers of data sets of relevance? Is imaging, so do you want to have images of viruses? No, probably not. Do you want to have images from research projects? So some of those, you probably couldn't see the thumbnails, but some of those EM images are events in viral life cycle invasion. So you want to have some of those, but it's not a regular assay. So sequencing is an assay, so we get very big numbers. But I think imaging on this occasion isn't an assay. But if some other disease or world event arose where imaging became the default assay that people use around the world, then I would expect the numbers to be much bigger. But if one decided to share the images, one may just decide to abstract something from them. So I don't know that we should necessarily expect so much, and I don't think that the numbers 29 is not necessarily a bad number. But I do suspect there are image-based studies that have produced useful, potentially reusable data that could be put into the system. But I think that the key thing is that there is, through the Bioimage Archive, there is now an indexing mechanism. We may need to extend that to index things beyond the Bioimage Archive, because I know that not everything goes there. But I guess that if there's a data set of relevance for COVID or other pathogens or infection biology, I think the key thing is that either there's a path to putting that into Bioimage Archive and tagging it appropriately so we can find it, or we find out what the connection should be. So I think the thing to do is, if there, I don't know. I think it's for your community. Your community has the expertise on what's being done and knows the studies and the data types. Would be if you come up with things that you feel should be represented and better integrated, then we should talk about them. For us, we're sort of, if they come into the Bioimage Archive, then we would, I hope, automatically pick them up. But I can appreciate that, this is a huge, complex community with all sorts of different systems, and not everything goes into one place. I fully appreciate that.
Yeah, thanks a lot, Guy. And I think we have good news that in Euro-BioImaging, we will soon have our first fair image data specialist, and that person will look exactly into this question. And then hopefully we can increase significantly the number of data sets indexed for the COVID-19 data portal, because they exist. It's just that it's not connected yet.
Yeah, excellent.
If the audience allows me a third question. So you presented the Global Biodata Coalition, and you're navigating a quite dynamic and complex landscape. You have the Research Data Alliance, you have the European Open Science Cloud, you have Alex here. Can you just tell us in a nutshell what the Global Biodata Coalition is different, or what they will do differently than the other international initiatives in this domain?
So the Global Biodata Coalition is about sustainability for data resources. So for those parts of the infrastructure that are serving up, preserving, making available data in some way, life science data in some way, the GBC is there to build sustainability, which is a process of adjusting and aligning the funding to different mechanisms globally, and aligning the data resources a little bit to that funding. So GBC is not a funder, but it's apart from those other organizations and initiatives you listed, because it's about building sustainability in data resources.
That's music to my ears, and I hope also to others here. Jonathan.
Yeah, hello, thanks for the presentation. I was just wondering, do you have any vision about what proportion of people who are looking for this type of data are actually coming through the COVID data portal? Is there any way of evaluating that?
No, I mean, this is sort of, this is how we're a little bit frustrated by our principles of making things open and anonymous. So what we show on the portal is either metadata that are fully open, in the case of controlled access data underneath, or it's full data sets that are fully open. And having a completely low friction entry point for people means that we don't want people to register, we don't want them to log in, we don't want them to, so we know very little. I mean, we can have a geographical distribution from IP addresses, and we can look at numbers and times that, that, so we can have the overall view. But often, because we don't try to retain any of the users, once they find the thing they're looking for, actually they often, in most cases, they get rerouted to a data resource somewhere else, external to us. And it could be that some of those data resources are looking at redirects and looking at provenance of users, but maybe they're not. But we're not systematically trying to control that or to monitor that. So actually, when you see an identifier cited from one of the underlying databases that we've been presenting that data set through the portal, we don't know whether they've got to it through the portal or not, and that's sort of frustrating because we have to justify what we're doing. But equally, it's a positive thing because they're finding the data somehow, and they're
But as you said, the data sources could, if you ask them, tell you what proportion of their traffic was coming from you.
Yeah, but only first time though. Only first time. So if I'm looking for an image data set, and then that gets me to, well, actually I know this, I can come directly here in future because I need lots of other image data. We wouldn't know about that, and they wouldn't know about that. So yeah, to an extent, we could do that. Yeah.
Okay. Thanks.
Thank you very much, Guy, for the answers to all our questions and the fantastic presentation and the overview. And Jonathan, maybe I can use this opportunity to introduce you as the coordinator of the EasyDoor project here. And I also see that we have more attendants here from Irina that are coordinating together as a team the EasyDoor project. And with this, I would like to hand over to my colleague Arina Rubina here from the Euro-BioImaging team. And Arina will briefly present you a few slides on the EasyDoor project, because this is an opportunity to capture new data for the COVID-19 data portal. Arina.
Yeah, thanks a lot, dear Antje, and thanks a lot, Guy, and also Jonathan for the discussion. So I really would like to use this opportunity. It's a very exciting one to get really some funding for research for scientists who are still interested in SARS-CoV-2 and COVID-19. So we heard that there might be some saturation plateau coming, but it's still quite acute, and Corona is still floating around. And Euro-BioImaging is really participating in this project as a strong partner and coordinating provision to imaging services and really open access. Next slide, please. To tell you briefly a bit about EasyDoor, so this is really a large consortium and a project funded by Horizon Europe, and it is led by Irina. We have a pleasure to have Jonathan here, and also Audrey, who is a scientific project manager, and we have also Dylan in the audience. And it really brings a large amount of service providers together from 32 countries, really representing a large spectrum of disciplines that allow people really to take services ranging from addressing characterization of pathogens to development of vaccines. And really the main goal is to provide free charge transnational access to the services across research infrastructures and networks. And the scientists should really get the support in their research on fighting epidemic and pandemic-prone pathogens and the outbreak. So there will be some links coming in the chat where you can check for more information. And the first call is open now, really specifically for SARS-CoV-2 and COVID-19. And of course, the project will last for 3 years, and more topics will be coming up. And we heard now about monkeypox already from Guy, so this will be also coming our way. And next slide, please. And I would like to advertise that Euro-BioImaging is really, on the one hand, coordinating the open access to imaging services, but also in this project provides advice on the roles of imaging services to fight infectious diseases and really to prevent or to address questions when there is some outbreak of pandemics. And Euro-BioImaging will also participate on fair data management. And together with Instruct, Euro-BioImaging is co-leading provision to analytical services. And if you would like to check out how to apply to Euro-BioImaging services and how to get open access, please check out the link. And you can also, if you have your mobile phone, you can also scan the picture and you will be redirected to our webpage. Next slide, please. And taking this opportunity, since EasyDoor is really a large consortium and there are really different services that are being offered really from in vitro studies to in vivo studies and really vaccine developments and even social science and epidemiology, then if you would like to learn more about it, there will be a webinar organized by Rna, and we have actually one of the speakers, Audrey, even in the audience, that will be taking place on 12th of July. So please, if you're interested in this initiative and in this opportunity to get funding to access services, please visit the website and register for this webinar. And with this, thank you very much for your attention. And I would like to hand over to Antje.
Thank you very much, Arina, for the quick overview on EasyDoor and transnational access funding opportunities for everyone. And with this, it's my pleasure to introduce Aastha Mathur, the Euro-BioImaging coordinator for image data services from our team, who will present the Bicovit project today, which is actually the brick between EasyDoor, then comes Bicovit, and then the COVID-19 data portal. So Aastha, you are the coordinator, as I said, of the image data services in Euro-BioImaging Hub. You have received your master's at the University of Heidelberg, followed by a PhD at EMBL in Heidelberg. After your postdoc at the Institut Curie in Paris, you have moved back to Heidelberg about a year ago to join the Euro-BioImaging team. Aastha has a background in bioinformatics, imaging, and image analysis. She's currently representing Euro-BioImaging, but also the European bioimaging community at the Eos Association in several EOS-related projects, including Eos Future and Bicovit. And Aastha is also coordinating the image data analysis services that are provided by the Euro-BioImaging nodes to all researchers that request them. So the stage is yours, Aastha.
Thank you very much, Antje. I'm gonna share my screen.
Perfect.
You see the screen, yes, and you hear me. All right, so thank you very much. And it was just already pretty great to hear so much about how data sharing and is an important process for combating a pandemic. And now we can try to look a bit at the kind of data that we are producing and how that can be beneficial in this process. So I'm going to share with you a couple of efforts that we're making in this direction. So first of all, image data and the pandemics. There are many different ways that image data can contribute to the pandemic research. And one of them could be from detailed things like structure of the viral particles, etc. And a lot of EM data has already been talked about. That is definitely a very important aspect to understand the viral particles and their spread. Then you can have data, large data sets from screenings, including drug screenings or siRNA screenings and so on, which can be very telling of what is going on or how cells respond to these viral particles, as well as going really down to the cellular mechanisms that relate to either the viral uptake or any of the responses that the cells might have due to infection due to viruses. And this is pretty much one of the reasons why imaging is an important data set that is included in the COVID-19 data portal. Now, as we've already talked about, for the data to eventually end up on the COVID-19 data portal, it first needs to go to this place, one of the open image data repositories, the Bioimage Archive. The Bioimage Archive is hosted by the EMBL-EBI, and it's a general purpose data repository which stores, indexes, and makes available all different kinds of bioimage data from all modalities. And the main objective there is to, first of all, make sure that to ensure reproducibility of results, as well as to have reference data sets, for example, large-scale atlas projects, etc. Almost to the degree of enabling research, which could include developing softwares or image analysis procedures based on this open image data, as well as data reuse. Now, the Bioimage Archive is basically the one place or the archive where all of the data can sit, but it also interacts very actively with other data resources, which are the so-called added value databases. One of the examples is, of course, the IDR, which is the house for reference data sets from imaging, as well as the Empire database, which holds electron microscopy data sets. So this is where the data can be. And what the general objective of this Bicovit project is to actually connect, to put more data into the Bioimage Archive so that it can then be discovered by the COVID-19 data portal and be exposed to all the people for potential reuse. Now, I'll take a couple of minutes to talk to you about a bit in details what the Bicovit project is covering. So the Bicovit project, or the Beyond COVID project, is actually getting data from very different and diverse resources. This could include national health data research infrastructures, hospitals. It includes different kinds of data, from basic research to biomolecular data, which could be related to patients or viral sequences. It could even include data from medical centers or electronic health records, as well as social sciences data, which includes often surveys about the patients. So it's really a diverse data set. And the first step there is definitely to mobilize the data, because the data sits in all these different places. After mobilization comes connecting very different kinds of data resources, trying to make sure that there is a way of connecting these data sets. Then to have standardized guidelines in terms of how to actually store this data, eventually with the aim of exposing this data for future analysis. And many different stakeholders can benefit from this, everybody from policymakers to health professionals to researchers from all over the globe. Now, talking about imaging data in this landscape, we are working mostly towards the far left of this pipeline, which is mobilizing the data sets in the first place. And to really fit it into what it benefits to us as a community, I would also briefly share with you another project that we are currently working on, which is the Eos Future Project. Now, the idea of this project in general is what we want to achieve is, first of all, to have this pipeline of data, this flow of data. The data can be acquired at different facilities, even with different research infrastructures, and this large and information-rich data would then need to be transferred into a standardized file format, and even the metadata needs to be standardized. After the standardization process comes workflows, which in this particular case of the Eos Future Project would be a cloud-compatible workflow, and eventually to be able to present this data in an open and fair image data storage. Now, we already talked a little bit about the Bioimage Archive, where this data would eventually need to reside, but these other processes, for example, the standardization process is going to be supported by our image data specialist, who's working on converting high content microscopy data into the OME SAR format. And this is in close collaboration also with the OME team. Once the data sets would be converted into these cloud-compatible file formats, the idea would be to do some proof of concept image analysis on it. Now, in this particular case, I'm not going to talk in details about this part where data, this was a part of one of the Eos Live demonstrator projects where the data was converted, open data was used through Galaxy pipeline for performing image analysis, and then eventually these results were available, and so was the workflow. Now, this full connection is then going to be made possible with Galaxy, and this is basically the Eos Future Project. And one very important place where Bicovit sits is the very first spot, which is to actually mobilize the data sets. And one of the reasons why we are attempting this is not just that we want to have, of course, it would be very beneficial to have a finished pipeline, but we also want to make it in a way that we are enabling people at every step of it to easily perform these processes. And hence we have a very modular data pipeline to enable eventually getting the data into the Bioimage Archive. So finally, what can we expect to get from the Bicovit project for ourselves? Well, one thing for sure is that we would probably get more fair image data sets into the Bioimage Archive, which in general would be beneficial to build future reuse cases on. What we can also get is general guidelines for verification, which will help us to, sorry, I missed that, which will help us to get more and more data sets into the Bioimage Archive, and in a format that it is actually conducive to reuse. The other advantage would be to really connect the image data to various different kinds of data sets. Like I mentioned already, that the Bicovit project aims to connect data sets from biomolecular data all the way to social sciences data. So it would be very beneficial for us to be able to connect to very different kinds of data sets so that others can actually benefit from the research that imaging scientists have performed. And as well as eventually the pandemic readiness, because once we have these steps, these guidelines, as well as these workflows in place, we are basically ready to incorporate new data sets quickly and in a proper fashion so that we can have more and more research based on these open image data sets. With that, I would actually quickly finish up and thank a lot the team that we're working with. So this is the Euro-BioImaging team, and on the side you see Jean-Karim Harish and Christian Tischer, who are experts at EMBL Heidelberg, who help us a lot, and Matthew Hartley, who's the team lead at the Bioimage Archive. And I thank you for your attention, and I'm happy to have some questions and discussion.